Now we are going to fourth step. The fourth step is nothing but collection of hits in a PDB format or SDF format. So if actually we have carried out GCMS analysis, and if you want to get the SDF structure of those molecules, then you have to go to this Pubchem databases. If you want to get the structure of drugs, then you should go to Drugbank databases. Okay, so as I told earlier, so if you carried out any GCMS analysis, if you want to get the information about these compounds, okay, then we have to go for Pubchem. So if you go to Pubchem databases, you can go and then you can give the name of the compounds. For example, here it is given like propanoic acid 2. So you can give the name like propanoic acid 2. So you will get a structure. So it is space now. Even though it is not giving, the 2 is unemployed. So just we give propanoic acid. So now you get the compound. Okay, so you click that compound. And then its structures will be available. And you can get the information about the compounds. Then you can download the structure of this particular compounds. So it's better to download this 3D conformers if it is available. Rather than taking 2D, it's better to use 3D conformers for the studies. So if you are carrying out GCMS analysis and you are preparing a list of chemicals, then you will get the structure of those compounds from the Pubchem. The second option is you can also use the therapeutic or the plant databases to collect the structures. We have already selected 4 hits, like this antrographoside, antrographanine, antrographolite. So these compound structures can be easily derived from this particular Indian medicinal plants databases. For this purpose, you can go here and in that first page, summary phase, if you go down, you will get the link for 3D structures. Okay, better to, if it is available PDB format, you can download. If the PDB format is not available, you download this SDF format. So you will collect this SDF format of the hits. Okay, so like this, you can collect the structure of all the hits. After downloading the hit structure, you can add the word, this particular antrographanine, to as a prefix to this particular molecule, so that you can easily identify which is the structure of the molecule. So like this, we can download all the SDF format of the drugs. Okay, so we can download all the SDF format of the drugs to this download folder, and we will rename it by using this link. In the home summary page, you can get the link for the download. And one more thing is that we can also copy this particular SMILE structure. Okay, so we can also copy the SMILE structure of the drugs and we can paste it in a notepad. So it might be used for that ADME-tox prediction step. So for that purpose, we are copying the SMILE. We are downloading the SDF format. So you can download the SDF format either from the plant databases or you can download the structure from the Pubchem. So now the structures are downloaded in an SDF format. But for docking, we need a PDB format. So for this conversion, similarly, if you want to get information from Drugbank, if you want to download drugs from Drugbank, then how you can do it? So you will go to Drugbank, and then in the Drugbank, you have to log in yourself. It needs a academic login. So you go to drug bank and log into it, and then you can select a target or drugs. If you know the name of the drug, you can give the drug, or if you know the target name, you can give the target name, and then you will search the database. You will get the list of target. From the target, you select the target and you can get the information about the drugs. You can see this is apoptosis regulator. BCL2 is a apoptosis regulator. You can see BCL2. So here we instead of giving space, better not giving the space, BCL2. Now you will get, see, apoptosis regulator. There are two outcomes. So this link we will go. So in this regulator, if you go downwards, you will see the list of drugs which will act against this particular target, BCL2. So from this we will select targets. So we select one of this link, and then and downstairs it will give a list. See here the pelitoclax, that is what the drugs we have listed. So we can check the drugs here. So we click this particular drug and we will get the SDF format, SMILE, as well as PDF format is also available. So if you want, you can get even the PDB format of the drugs straight away from here, or you can get the SDF format and then you convert the SDF format into PDB file. So you select this SDF format and then you save this file. Okay, so now to save this file in the download. So now paclitaxel is ready, and we also need one more drug, that is, it is called venetoclax. Similarly, we can also collect the SMILE structure of this drug by using this SMILES option. Okay, so you can download the SMILE structure, and you can copy that SMILE structure and you have to save it in the notepad. So I have already collected and stored. Now it is a drugs, so you give this name of the drug and you select this option as a drug, and you can again get the information about the drug, and you can download the structure of the drug in a SDF format, or if you want, you can download the structure in a PDB format. So you can save the file. So to understand easily, the name of the drugs can be added as a prefix, so that we can easily identify the, so 581 is going to be the venetoclax. Okay, so you rename it so that we can easily identify the drug. Similarly, you can also add the paclitaxel name to this particular drug. So it's all used to identify them easily. For that purpose only we are adding these things. Okay. So drugs downloaded, ligands download. Like this, we will download all other 4 ligand structure in SDF format with the help of this IMPPAT databases, and we also collect the SMILE structure. Okay, so this is how we have prepared the hit, that is the ligand list. And now we have selected the ligands and we collected the SDF formats, but for docking purpose we need a PDB format. So we have to convert this SDF format into a PDB format. So for that purpose we will use the Babel. So open source Babel is a online tool which is available. We can also download the software and we can install it. So we will use this particular Open Babel server. Okay, so this is all. So how to convert the SDF format? We have to open this SDF format in a notepad. Then you Control A, Control C, to copy and then paste that file here. Once you paste the file, you can see the structure loaded here. And then the input structure what we are providing is SDF format. So select the SDF format, and what the output format we need, PDB format. So select PDB as an option, protein data bank, PDB, protein data bank format. Once you select all these two formats, you click the convert option. So now the file is converted, and then you can select Control A, Control C, and then you can save this file as a new, open new WordPad and paste it and save as. Again you select this is an all file, and we know this is the drug we selected is palidoxal, so we will use the same name for this file, but the file format is PDB. So you give this PDB and then you save it. So now the SDF file format is converted into PDB format. So like this we will convert all the drug structures from SDF to PDB, ligand structure from SDF to PDB. So like this you convert all the structure. Once again, if you want to convert the other drug, that is, for example, the one more drug is venetoclax. So you can open the structure of this. So we copy the SDF format and then we paste the format, and then when the structure is loaded, you convert this SDF to, you select SDF, output is PDB, okay, then you use the converter option. The file is converted, you copy Control A, Control C, and you can create a new file, and then you save the file by using the save as option. For renaming it, use the same name with the PDB exception. Convert this file as all files option, so you can save. So the drugs are converted from SDF file to PDB file. So like this we can also convert the other ligands also from SDF to PDB file by using this OpenBabel software. So now we have completed the selection of disease and target hits list, and then we downloaded the PDB structure and prepared for docking, and then fourth we downloaded the ligands and we converted SDF to PDB format for docking. So 4 steps of docking were completed.